Jen Marini, you may turn off your camera. Okay, so anyway, good day everyone. Good day everyone. So this is now the last topic for our first quiz in the midterm, the labeled immunoassays. Okay, buti pa siya may label. So that's my introduction for labeled immunoassay. So uh, we have to go back to the basic principle of antigen and antibody complex. So this is an antigen, and then this is the antibody. So whenever there is a reaction, we call it immune complex. However, we cannot see or we cannot visualize and we cannot measure immune complex. So we cannot visualize the immune complex. We cannot measure the immune complex unless we will be using something to measure it. This is where labeling will come into. So there are actually three major types of labels. Okay, so these particular labels will serve as an indicator. So they will serve as an indicator so that we can visualize and measure this immune complex. So these particular indicators or label can be in the form of enzyme. It can be in the form of radioisotopes. It can be in the form of fluorescent dyes. And the fourth one is the newest. This is the chemiluminescent indicator. So these are the possible indicators that we can use for labeled immunoassays. Okay, so in this particular chapter, we will be discussing the format for labeled assays, the heterogeneous and homogeneous assays, radioimmunoassays, enzyme immunoassay, fluorescent immunoassays, and chemiluminescent assays. Now, if you are wondering what is this particular photograph is all about, this is actually the automated ELISA reader, which, by the way, our school acquired, and you will be the first batch to utilize it in our limited face-to-face. So the ELISA reader can actually measure simultaneously 96 patients if you are using a 96 well, 96 samples if you are using a 96 well type, no? So this is one of the things that I am very most excited about in our limited face-to-face, particularly for the finals, no? The automated ELISA reader. But nonetheless, for this particular lecture, I will be, of course, discussing the formats for labeled assays and what's the difference between the heterogeneous and homogeneous assays, and then the radioimmunoassays, the enzyme immunoassays, the fluorescent immunoassays, and then the chemiluminescent assays. So labeled immunoassays are designed for antigens and antibodies that may be small in size, okay, or present in a very low concentration. So they are present in a very low concentration. And for us to be able to measure it, even if they are in a very low concentration, that talks about specificity, sensitivity, sorry. So that talks about sensitivity. A sensitivity method, as a highly sensitive method, would have the ability to measure and analyze even if they are present in a very small size or in a very low concentration. So what are these analytes that are present in a very small size? So this could be bacterial antigens, this could be hormones, this could be drugs, especially if you're using it for therapeutic drug monitoring. This could be tumor markers, and this could be specific immunoglobulins or antibodies. So they can be measured using the labeled immunoassays. So we are using labeled reactants to monitor the amount of specific binding that has taken place. So when you say specific binding, again, we are referring to the immune complex. So when you say immune complex, there is a complex between an antigen and antibody. And this particular complex can only be measured if we are using a labeled reactants. So by the way, you might get confused. How about hormones and drugs? Now, in the context of labeled immunoassays, we will be treating hormones and drugs as antigen. So these hormone drugs and even tumor markers will be treated as antigens in the context of labeled immunoassay. So there are actually two types of immunoassays. So we can possibly use the competitive immunoassays, and the other one is the non-competitive immunoassays. So let's talk about first the competitive immunoassays. Okay, the first step here is that all the reactants are mixed together simultaneously. So when you say all the reactants, okay, this includes all the antigen and antibody. So they will be mixed together simultaneously. Okay, so there is a competition. Okay, so the labeled antigen competes with patient's unlabeled antigen. So there are two types of antigens here, huh? Okay, the one that comes from the patient and the one that comes from the reagents. Okay, so this is unlabeled, and the reagent's antigen is the labeled antigen. Okay, so they are competing for what? They are competing for limited antibody binding site. So this is the limited antibody binding site. Okay, so let's make an analogy. So konti lang yung mga babae, mas madami yung mga lalaki. So yung mga lalaki, they are competing for a limited number of ladies. So that could be an analogy that we can use. Okay, so now the reagent is labeled antigen. Labeled with what? Okay, this antigen can be labeled with enzyme, that is for enzyme immunoassay, or this can be labeled with radioisotope. And if they're labeled with radioisotope, this being used in radioimmunoassay. Or this can be labeled with fluorescent dyes. If it's labeled with fluorescent dyes, then we're using it for fluorescent assay. So the amount bound label is inversely proportional to the concentration of labeled antigen. Meaning to say, if there are more labeled antigen that goes here, meaning to say the patient's unlabeled antigen is actually low in concentration. So they are inversely proportional. So to give you an idea, so we have here two scenario, no? Okay, so we have here two scenario. The first scenario is sample A, and then the second scenario is sample B. A known concentration of analyte in patient sample. So the red dots is actually the patient's sample. Let's say, for example, the red dot is HCG. It's a hormone, human chorionic gonadotropin. Okay. So the human chorionic gonadotropin in this particular example will become what? This is our antigen. Okay, this is our antigen. Okay. And then the yellow stars, the one that you see here, this is the labeled antigen. And the labeled antigen comes from the reagents. It comes from the manufacturer. Okay, let's say for sample A, the patient is not pregnant. For sample B, the patient is pregnant. Okay. So you now have two types of HCG. Okay, the HCG here is HCG coming from the patient sample. That's your first type of HCG. And then the second HCG is the HCG labeled. So let's say, for example, we label the HCG with an enzyme labeled HCG. Okay, so what will happen here in sample B, since the patient is pregnant, the patient's HCG and the HCG labeled antigen with enzyme, or the enzyme labeled HCG, will compete for a limited antibody binding site. So let's say that these are anti-HCG antibodies. So compare patients who are not pregnant and compare patients who are pregnant. So for patients who are not pregnant, you only have the labeled antigen, HCG, enzyme labeled antigen, and they will bind freely. Into the anti-HCG. However, for patient B is a pregnant sample. Okay, so what will happen? The HCG will combine for a limited binding site. So they will compete for a limited binding site. So what will happen here? We allow, so this is now your enzyme labeled HCG. They can freely bind to anti-HCG. Whereas for a positive patient, you have the enzyme labeled HCG plus you have the patient's HCG. And we wash it, and those antigens that are unbound will be removed. So wash to remove unbound antigen. And then we add substrate. So a substrate is actually substance being acted upon by the enzyme, and adding the substrate will have color reaction. Okay, so compare patient A from patient B. The color here is more intense color, and then the color B here is less intense. And the reason why it is more intense because of high signal. Why is it high signal? Because we have more enzyme that is bound to anti-HCG. However, for patient B, it is less intense. Less intense because there is a less signal. And the reason why it has less signal because less antigen, less enzyme labeled antigen has actually bound to anti-HCG. So the thing here is that for competitive binding, the signal strength is usually inversely related to the analyte concentration. What does it mean? The higher the signal, the lesser the analyte concentration. The lower the signal, the higher the analyte concentration. In our example, patient A, let's take a look at patient A. . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . Patient A has higher signal because patient A doesn't have HCG. Patient B has less signal because patient B has more HCG. And who is pregnant? Is it patient A or patient B? So patient B is pregnant. Okay, so this is now the principle of competitive binding. Okay, so that is the principle of competitive binding. Okay. Might run out of battery. So I hope you understand the principle of competitive binding, competitive immunoassay. Okay, now this is now the non-competitive immunoassay. So the antibody is first absorbed to a solid phase. So a solid phase could be a plate or it could be a tube. So the antibody is first absorbed into a solid phase. And then the unknown patient is allowed to be captured. So this is now your antibody, and then this is now your antigen. Okay. After washing, so unbound antigen will be removed, and then the second antibody is added. So this is the simple way of illustrating the non-competitive immunoassay. So there is no competition for a limited antibody binding site for non-competitive immunoassays. So therefore, in non-competitive immunoassay, the amount of label measured is directly proportional to the amount of patient antigen. So in non-competitive immunoassay, for example, the more intense the color, then the more patient antigen is present. Okay, so what are the possible labels that we can measure, that we can utilize? We can utilize radioactive substances, we can utilize enzymes, we can utilize fluorescent dyes or fluorescent compounds, we can utilize chemiluminescent substances. All of these, okay, are actually being used as labels or indicators to determine the reaction. Okay, so there you have it, no? The difference between non-competitive and competitive immunoassay. So let's apply. Okay, so sample A and sample B. So sample A is negative for analyte, and sample B is positive for analyte. Okay, so what will happen here? Let's say, for example, this is um, the antigen test for COVID using non-competitive ELISA. Okay. So what are the things? So this is the solid phase. Okay. So the one that is being placed in the solid phase, is antibodies. So antibodies against COVID-19. So let's say that the patient has viral proteins. So what will happen for a positive patient? The viral proteins will go to the antibodies. And if the patient is negative, there is no viral proteins. Okay? And then here, you will now be adding enzyme labeled immunoassay, enzyme labeled antibody. So enzyme labeled antibody. How does it look like? Looks like this, no? There is an antibody, and it has an enzyme at the Fc portion. So there is an enzyme. So something like this, no? There is an enzyme at the Fc portion. So what will happen is that the antibody, which is also specific for COVID-19 SARS-CoV-2, will bind. And then we add substrate. So there will be a change in color. So what will happen here is that the more intense the color, this is intense the color, then it will result into increase in signal. And increase in signal means that more antigen is present. So this is an example of a positive patient for SARS-CoV-2. Now, sample A is negative for SARS-CoV-2. So again, no immune complex is formed. That's the reason why there's no enzyme-labeled antibody will be just washed away because there's no antigen here. Okay, so you'll not be measure anything at all. So this will be the graph. Okay, this will be the graph, no? Okay, so this is if we are detecting, if antigen is being detected. If antigen is being detected. Now, antibody capture is also possible. So antibody capture is also possible. So antibody capture means that you are doing antibody testing. So the principle is the same, except that in the solid phase, instead of placing antibody, what you will be placing here at the solid phase is an antigen. And then patient's antibody will be added, and then the so what's the difference between the enzyme here and enzyme here? The enzyme here is now the anti-human enzyme labeled anti-human antibody. Because the enzyme labeled anti-human antibody, as you can see here, will bind at the Fc portion of patient's antibody. Okay? So if you are detecting antigen, what you will use is the enzyme labeled anti-human antibody. If you are detecting antibodies, let me correct myself again. If you are trying to detect antibodies, such as the one that we illustrate here in the third row, you will be using an enzyme labeled anti-human antibody. But if you are detecting an antigen, you will simply use an enzyme labeled antibody. Okay, so that's the basic difference. So basically, this is the non-competitive immunoassay. So I hope you do understand that. Okay. So let's differentiate now the characteristic of competitive and non-competitive immunoassay. So here, all reactants are mixed together. Here, it is either the antibody or antigen is absorbed to the solid phase. In competitive, like what I've told you, labeled antigen competes with an unlabeled patient antigen for a limited number of antibody binding sites. Here, unknown patient antigen reacts and captured by the antibody. So unbound antigen are removed by washing, then a second antibody with a label is added. So basically, here's the difference, no? In competitive, bound label is inversely proportional to the concentration of the labeled antigen. But in non-competitive, the amount of label measured is directly proportional to the amount of patient's antigen. So here, the more intense the color, then the more antigen is present. Here naman, the less intense the color, then the less antigen is present. But it says also here in the table, in both types of assay, whether we are talking about competitive or non-competitive immunoassay, the label must not alter the reactivity of the molecule, and it should remain stable for the shelf life of the reagent. Okay, so that's the difference between the competitive and non-competitive immunoassay. Also, antibodies that we should be using should have high affinity and very specific for the antigen. See, because we do not want, so this is very important so that we can prevent false positive result. And then standards or calibrators. So when we say standard, it is a substance of known concentration. Okay. And then unlabeled analytes should also be present. Separation methods include a solid phase vehicle for separation. So that's the reason why in our ELISA washer present in our lab, there's a machine that would do the separation phase. Okay, so that we could ensure that specific binding takes place, complexes are formed and remain attached to the solid phase. And then detection of the label. How do we detect label? If we are using enzyme, if we are using enzyme, the label methods of detection. Photoelectric method, such as a spectrophotometer. If you are using a radioisotope, it can be detected using a scintillation or gamma counter. If you're using a fluorescent dye, then it can be detected using a fluorescent microscopes. So these are the machines that should be present in our laboratory. So I'm so proud that for us in EAC, we have the ELISA washer reader. So we have the ELISA washer reader machine that can detect any enzymatic reaction that has taken place. You have to see that. Okay, so let's talk about the first type of immunoassay. Okay, so the first type of immunoassay, the first one that was developed, okay, is the radioimmunoassay. Okay, so again, it utilizes radioactive labels. So there are several examples of labels that can be utilized, but the most popular is the isotope 125. Okay, so isotope 125 is the most popular, no? So it emits gamma radiation, which can be detected using a scintillation or a gamma counter. It is extremely sensitive and precise and can even measure trace amount. So when we say trace amount, we are referring to a very small amount of hormones, serum proteins, and even vitamins. So the immune complex would appear like this, no? So the immune complex, this is the patient's antibody. Okay, this is the patient's antibody. This is now the antigen. So it could be in the form of antigen. In this context, could be a vitamin, a hormone, and then this is now the radioisotope. So the immune complex, so radioisotope will be attached here to the antigen, and this one would be measured in a gamma counter. So let's have two scenarios. So the first scenario, so you have the radio-labeled antigen, and here, so radioimmunoassay is considered to be as competitive, no? Because patient's antigen and radio-labeled antigen competes for specific binding sites. So after washing, those that are unbound will be removed, and then this is measured. Since you have here patient's antigen, so there would be signal that is measured by the gamma counter. So signal will be measured by a gamma counter. So what if let's compare now two scenarios, no? Patient 1 and patient 2. So patient 1 has less patient's antigen, and then patient 2 has more antigen. So what will happen? So antigen will bind, more patient's antigen bind here at the limited binding site. because there are more patients antigen for sample number 2. So will happen. This one would have um, this one would have actually more signal as compared to the other one. So sorry, this one would have low signal. This one will have high signal. Pag sinabi nating high signal, the radioactivity is high. Pag sinabi nating low signal, the radioactivity is low, which is detected at the gamma counter. So high signal means what? Less patients analyte or antigen. Low signal means more patients analyte or antigen. Okay, so that's the basic principle of radioimmunoassay. So this is an example of competitive binding. So this in the radioactivity is indirectly proportional to the amount of patient's antigen present. Okay, so yeah. So this is how a gamma counter look like. So scintillation counter, the sample will be detected here, and then there would be signal indicating radioactivity. So unfortunately, we do not have this in our laboratory, but do you know that medical technologies has another career opportunity? So some medical technologies can even work in nuclear medicine laboratory. So in nuclear medicine laboratory, you can actually work as a medical technologist and do procedures such as radioimmunoassay. So again, remember again, the amount of label in the bound phase is indirectly proportional to the amount of patient antigen present. But these are the disadvantages. Of course, working with radioactive substances, it could be physically dangerous. But do you know that I have friends who are working nuclear medicine, and they're saying that the radioactive isotopes that they are dealing with is actually much lesser than the radioisotope that we are encountering in our daily basis. Let's say, for example, in old television set, not the television set, not the TV monitor that we have nowadays, but in the old days, they have TV with picture tube, and it emits radioactive substances. But now we are using smart TV, so fortunately, they don't emit any more radioactive substances. Disposal of low-level radioactive waste. So you should have a mechanism on how to dispose iodine 25. Okay, and then of course short shelf life of some of the reagents. So these are the disadvantages of using radio immunoassay. Okay, so let's now discuss the enzyme immunoassays. Okay, so when we say enzyme immunoassays, so instead of using radioisotope as our label, here we are using enzymes as our labels, which reacts with suitable substrates to produce breakdown or products that may be chromogenic, fluorogenic, or luminescent. So what's the common denominator of this? Common denominator is there would be increase in the intensity of color, and an increase in the intensity of color will serve as the signal. And what are the possible enzymes that we can use? So we could use alkaline phosphatase and horseradish peroxidase, or may also add beta galactosidase. So these are the possible enzymes that we can use for enzyme immunoassay. So this one have the highest turnover. So when you say turnover, the concentration of substrate rates, it's very sensitive and easily detected. So among the three, or among the four, radioimmunoassay, enzyme immunoassay, fluorescent immunoassay, and chemiluminescent assay, the one that is very common because it's easier to perform and it is not as expensive is the enzyme immunoassay. So that's the reason why it's the one that we can afford in our laboratory. But then again, we're lucky because not all schools have the ELISA reader. Okay, so the principle is like this, no? Um, antibody will actually go antibody in the solid phase will bind with antigen, and enzyme labeled antibody will bind with another would bind with the antigen. So there would be a sandwich phenomenon, no? So something like a sandwich because antigen is bound in between these two antibodies. So the first antibody is the one that is bound to the solid phase, and the second antibody is the one labeled with enzymes. Okay, there are two types of ELISA. Okay, so let's talk about it, no? So the first type that we will be discussing here is called the heterogeneous enzyme immunoassays. Okay, and the other one is called the homogeneous enzyme immunoassay. Okay, so let's talk about first the heterogeneous enzyme immunoassay. This is the most important requirement. Okay, this is the most important requirement of heterogeneous enzyme immunoassay. It requires a step to physically separate free analyte from bound analyte. Okay, free analyte came from the patient sample. Bound analyte came from the manufacturer. So it is a part of the reagent. And when we say bound analyte, sabihin, it is the enzyme labeled analyte. Okay. So heterogeneous enzyme immunoassay, therefore, will also use the principle of competitive assay. So this is now our second scenario wherein we are discussing competitive assay. So by now, you now have the idea what do I mean. So when I say competitive assay, it involves competition, okay, between enzyme labeled antigen competing with unlabeled patient antigen for a limited antibody binding site. So competitive assay therefore is utilized for measuring small antigens that are relatively pure, such as drugs and hormones. And the good thing about it is that this one have a very high specificity. But then the most important requirement is that you should be able to separate physically, literally, free analyte from that of the bound analyte. So there is also a non-competitive enzyme immunoassay. So the first heterogeneous immunoassay that I've discussed here is the competitive assay. Okay? And then the second one is the non-competitive enzyme immunoassay. So the non-competitive enzyme immunoassay is very practical because it will offer high sensitivity, specificity, simplicity, and low cost. So the most common example of non-competitive enzyme immunoassay is ELISA. And ELISA is not a name in this case, as ELISA stands for Enzyme Linked Immunosorbent Assay. Okay? The one of the most common immunoassay in the laboratory is ELISA. And it is being used to measure antibody production to infectious agents that are difficult to isolate for autoantibody testing. So we are using it, for example, for detection of antibody against HIV, Hep B, and hepatitis C. And this is also the principle for the point of care testing, POCT, and even the home testing. Okay, so if you remember, so we now have rapid antibody test, rapid antigen test, urine pregnancy test. All of these would actually employ the principle of non-competitive enzyme immunoassay. So let's talk about the heterogeneous ELISA. Okay, enzyme immunoassay. So ELISA. And ELISA, antigen is bound to a solid phase. And then patient serum with a known antibody is added and we give time to react. So in this particular case, we are detecting antibodies from patient sample. Now I want to give emphasis to this one, guys. If I say that we are detecting antibodies for patient sample, the most important and the most common specimen is a serum. Okay? So in ELISA, antigen is first added to the solid phase, okay? And a known antibody is added and given time to react. And then, after a wash step, an enzyme labeled antiglobulin or immunoglobulin, this is the secondary antibody is being added. So the second antibody react with any patient antibody that is bound to the solid phase. So let me draw this. I'll try to draw. So, oh, sorry. Yan. So this is the solid phase. This is the antigen. And then the patient antibody is bound here. And then we will now be adding so the patient antibody is the first antibody. And the second antibody is the enzyme labeled anti-human globulin, which will bind at the Fc portion of patient's antibody. Then we add substrate. After adding substrate, there will be what? Changes in color. The more intense the color, then the more patient's antibody is present in the solution. Okay, so that's the basic principle. That's the basic principle of heterogeneous ELISA. So if no patient antibody is bound to the solid phase, the second labeled antibody will not bind. After a second wash, enzyme substrate is added. So the amount of color, fluorescence, or luminescence is measured, and this amount is directly proportional to the amount of patient antibody in the serum. So let's say in a negative sample, you have an antigen, but no patient antibody. So even if you will be adding antiglobulin, that is enzyme labeled, this one will not bind to the patient's antigen because this antiglobulin fluorescent labeled antiglobulin will only bind at the Fc portion of patient's antibody. So this is how it is designed. It will only bind to the Fc portion of patient's antibody. So the more intense the color, the luminescence, then that's directly proportional to the amount of patient's antigen. So something like this, no? So antigen is bound to the solid phase, and then patient's antibody is added. And then this is now the antiglobulin, enzyme labeled antiglobulin. And then substrate is added, and that will result in the color change. Okay, so this is the principle of indirect enzyme immunoassay. So what is the purpose of the indirect enzyme immunoassay? Okay, so the purpose is to detect Patient's antibody. Let me correct this one. This is not a patient antigen, but it is the antigen bound to solid phase. So this is the antigen bound to the solid phase. Okay. So we also have what you call the capture assay, which is also known as the sandwich immunoassay. So the sandwich immunoassay are best suited to antigens that have multiple determinants. Okay, so it can detect antibodies, cytokines, proteins, tumor markers, microorganisms, particularly viruses, and it is also being used in the measurement of immunoglobulins. So what will happen here is that excess antibody attached to the solid phase is allowed to combine with the test sample to capture any antigen that is present. So let's say, for example, this is the solid phase. A while ago, antigen is in the solid phase, but now it is the antibody. Antibody is now in the solid phase. So it is designed to capture antigen. After an appropriate incubation period, enzyme-labeled antibody is attached. Again, kanina, we added antiglobulin, enzyme-labeled antiglobulin, which will bind at the Fc portion of the patient's antibody. But this time, we are adding enzyme-labeled antibody. Okay? The second antibody recognized a different epitope or binding site than the solid phase antibody. And now we have now completed the sandwich. So therefore, this is now a sandwich phenomenon. Why sandwich? Because you have here the first antibody, you have here the antigen, and then you have here the second antibody. So two antibodies sandwich an antigen. So that's the reason why it's called the sandwich phenomenon. So in heterogeneous enzyme immunoassays, particularly for the capture assays, either a colored or chemiluminescent reaction product is detected. So the enzymatic activity is directly proportional to the amount of the antigen present in the test sample, meaning the more intense the color is, then the more antigen is present. So enzymatic activity is directly proportional to the amount of antigen. So again, it's another way of showing the sandwich. So this is the solid phase. So this is the first antibody. This is the antigen. And yung red, this is the second antibody. So again, there is a what you call a sandwich phenomenon. Okay, a while ago, we're talking about heterogeneous enzyme immunoassay, wherein washing is required. But in this case, we're talking about the homogeneous enzyme immunoassay. In homogeneous enzyme immunoassay, this particular procedure do not require a washing or a separation step. Okay, so enzyme activity is also directly proportional to the concentration of patient antigen or HAP10 present in the solution. So no washing, and when antibody binds to specific determinant sites on the antigen, the active site on the enzyme is blocked, resulting in a measurable loss of activity. So homogeneous assay, however, is less sensitive than heterogeneous, but they are actually more rapid and simple. So we will also be doing this in the limited face-to-face. So it is being used to determine low molecular weight analysis of serum and urine, hormones, therapeutic drugs, and drug of abuse. So for example, I have here, cannot open my cabinet. I have here an example of rapid antigen test. So this one is an example of homogeneous assay. So the one that you can, the DIY, no? Yan. So yeah, yung rapid immunoassays. This one is very common. So in our laboratory, We have rapid immunoassay for HIV. We have rapid immunoassay for syphilis. We have rapid immunoassay for Hep B. Okay, so they are membrane-based single-use disposable assay. So it involves antigen or antibody being coupled to the membrane, and it's very simple to read, no? You don't need a machine to read. Just need your eyes. And all you have to do is to look for a color reaction. Though it may require separate addition of patient sample, and then the wash reagent or the buffer, the labeled antigen, and then the substrate. So let me open the kit, no? So yung kit, merong swab kasi ito yung pang-collect ng patient's antigen. And then this is the cassette. This is the cassette. And then this is the example of buffer solution, no? Yung buffer solution. And this is the buffer solution. So this is the buffer solution will serve as the wash reagent. Yan. So in this particular example, so you have to add clinical sample with analyte here. Okay. So what's the principle? In the conjugate pad, you have monoclonal antibodies with colloidal gold. So gold will serve as the indicator so that you'll be able to see a positive reaction. So if the patient is positive with antigen, as the one that you can see here, it will bind with monoclonal antibodies with antigen. Then it will now go to the lines. The first line that you have is a positive line, and the second one is the control line. Okay? So if in the test line, okay, if the sample is positive for patient antigen and it passes through the test line, it means that the patient antigen were able to bind with the monoclonal antibodies here, and monoclonal antibodies are also present. So these monoclonal antibodies are very specific. So let's say, for example, this is a rapid antigen test for COVID-19. So here is the anti-SARS-CoV-2. This one is also an anti-SARS-CoV-2 with gold. So the analyte that you're able to do here is also a sandwich phenomenon, because here you have the first antibody, and then this is the antigen, and then this is the second antibody. But since you pass through the test line, you are able to pass through the test line, this will cause colored reaction in the test line. So that's positive. Okay. Now, some of the excess will pass through the second test line. This is the control line. So the control line will also consist of anti-IgG monoclonal antibodies. This is like the anti-globulin monoclonal antibodies. So the anti-globulin monoclonal antibodies will bind at the Fc portion of the first antibody. This is the first antibody. This is the second antibody. So meron siyang Fc portion, so dito magbabind. So magkakaroon din ng line. Magkakaroon din ng color dito. So in fact, it's very important that there is a color in the control line to check that the result is valid. Okay, so ganito yung nangyari tuloy, no? So this is the control line na merong red. Yung test line, walang red. So ibig sabihin, yung patient na to, walang COVID-19. Wala siyang SARS-CoV-2 antigen. Pero kung may SARS-CoV-2 antigen siya, dadaan siya dito sa test line. Yung test line, meron siyang, di ba? Meron siyang antibody. Meron siyang monoclonal antibodies na specific dun sa antigen. Di ba? So, pag dumaan siya rito, katulad ng example natin kanina, so monoclonal antibodies plus patient antigen plus monoclonal plus antibodies with gold. So, magsa-sandwich siya, then magkakaroon ng line dito. So, that's the basic principle of rapid immunoassay or immunochromatography assay. Para hindi malito, we should be able to enumerate. Okay? So, the component, the analyte, is the substance of interest. Ano yung example natin? The SARS-CoV-2 antigen. Okay? And then, the second component, antibody with colloidal gold. So, ano yung example natin dito? This is the anti-SARS-CoV-2 antibody with gold. Ano yung purpose ng gold? It serves as the indicator. And the third analyte is the anti-IgG or antiglobulin. Okay, ano yung purpose ng antiglobulin? The first antibody, it will bind at the Fc portion of the first antibody. Okay, so that's the purpose. Okay, so that's the purpose of the third component. Okay, so rapid immunoassay. Okay, so let's talk about fluorescent immunoassay. So this one is restricted to qualitative observation. So present or absent. Unlike ELISA and radioimmunoassay, you can measure the concentration. Here, you'll just say it's positive or negative. And it involves grading the amount of fluorescence against a dark background. So you can have +4, +0, just grade it. It is used for rapid identification of microorganisms in cell culture or infected tissue. If it's found in the infected tissue, the procedure is called FISH, fluorescent in situ hybridization, which we will be discussing in another chapter. Tumor-specific antigen, neoplastic tissues, and transplantation antigen. So the fluorescent immunoassay can be of two types. Okay, so the first one is the direct immunofluorescent assay, and the other one is the indirect immunofluorescent assay. So for the direct immunofluorescent assay, this is for the detection of antigen. So antibody that is conjugated with a fluorescent tag is added directly to the unknown antigen that is fixed to a microscope slide. So after incubation in a wash step, you have to use a fluorescent microscope. So this is not an example of fluorescent microscope. I just placed the picture here for artistic purposes, but that is not an example of a fluorescent microscope. So after that, you'll be able to see organism that glows in the dark. Okay, so it's a technique that will detect or demonstrate the presence of pathogen in a patient sample. So tulad nga ng sinabi ko, the direct immunofluorescent assay will detect antigen. So in the slide, which serves as the solid phase, so you place an antigen here, and then you add fluorescent labeled antibody. Okay, so what are the fluorescent dyes that you can use? So you can use fluorescein dye, or you can use The complete name of that is fluorescent isothiocyanate, or you can use a rhodamine dye. So there are several examples of fluorescent compounds that you can use in the fluorescent assay. So this is how it looks like, no? So organisms will glow in the dark using a fluorescent microscope. Okay, that is very intense, using an ultraviolet light. So it will only glow in the presence of UV light. The other one is the indirect immunofluorescent assay. So this time, patient serum is incubated with a known antigen attached to a solid phase, which means that this is one is used for the detection of patient's antibody. The slide is washed, and then anti-human immune globulin containing a fluorescent tag is added. So again, the anti-human immunoglobulin will attach at the Fc portion of patient's antibody. Okay. The most common application, so there would be some sort of a sandwich. So the most common application of fluorescent immunoassay is the FTA-ABS, which stands for fluorescent labeled treponemal antibody absorption. So it is a treponemal test, an example of a very sensitive treponemal test for syphilis. So that's the most common application. That's the most common application of indirect immunofluorescent assay. So we call it the FTA-ABS. So in FTA-ABS, we have a killed treponema pallidum, which is attached to the solid phase. And then we add patient's antibody that will attach now to the epitope of the treponema pallidum. Then we add fluorescent labeled anti-human globulin, which will bind at the Fc portion of the patient's antibody. Then there will now be change in color. So change in color. And if there's a change color, then that means that the person is positive for patient's antibody. So that person is positive for syphilis. Okay, so that is FTA-ABS. Again, FTA-ABS stands for fluorescent treponemal antibody absorption. It is an example of a treponemal test for syphilis in which the etiologic agent is treponema pallidum. So again, the difference between direct and indirect immunofluorescent assay. In direct, we are detecting patient antigen, and we are using a fluorescent labeled antibody, which will attach to the patient antigen. For the indirect immunofluorescent assay, we are detecting patient's antibody, and we are adding a fluorescent labeled anti-human immunoglobulin or anti-globulin, which will attach at the Fc portion of the patient's antibody. Okay, so that's the basic difference between the direct and indirect immunofluorescent assay. So there's another twist to our fluorescent immunoassay, and we call it the FPIA. FPIA is used to determine the concentrations of therapeutic drugs and hormones. And it is based on the change in polarization of fluorescent light emitted from a labeled molecule when it is bound by antibody. Okay? So if the labeled molecule is bound to the antibody, a molecule emits an increased amount of polarized light. So the more polarized light that is present, then the antibody. To give an example, okay, ito yun, ha? So you have two scenarios for FPIA. Okay? no toxin, and with toxin. Okay. Patient A is negative. Patient B is positive for toxin. Okay. So you now have the antibody. Since for patient A doesn't have antibody, so there is no immune complex. And then you will now be adding a fluorescent dye. Fluorescent dye. And since you are adding a fluorescent dye, this particular fluorescent dye would have more tracer bound and then lower rate of rotation. Because lower rate of rotation, meaning to say, mas mabilis mag-rotate because there is no toxin. And since there is a lower rate of rotation, so the polarization would be very high. Whereas scenario, in this particular scenario, toxin is present. So toxin will bind to antitoxin. And then if we will be adding a fluorescent dye will not anymore be able to bind to the antibody because of the presence of toxin. So these are less tracer bound, but the rotation is higher. And if the rotation is higher, then there is a lower polarization. So what's the lesson here? Okay, higher polarization means that negative, it is a negative or low results. Lower polarization, it is a positive result or with high concentration. So that is FTIA. So, so more, so yung polarization is actually, the rotation of the light is inversely proportional to the polarization, yung pag-ikot. Okay? So the last immunoassay that we have, before I dismiss you, this is the chemiluminescent immunoassay. Why do we call it chemiluminescent? The reason why we call it chemiluminescent is because it involves the emission of light caused by a chemical reaction, and this chemical reaction is typically known as the oxidation reaction, producing unexcited molecules that decays back to its original ground state. So the most common chemiluminescent substance that we are using in these assays are the following. So we can use luminol, acridinium esters, ruthenium derivatives, and nitrophenyloxylates. So these are the most common chemiluminescent substances. When these substances are oxidized, intermediates are produced that are of higher energy states. So upon reaching the higher energy states, these intermediates spontaneously return to the original state, giving off energy in the form of light. From high energy, it will go down to the original state, and as it goes down, it produces light. So chemiluminescent immunoassay can either utilize competitive assay. Competitive assay is being used for very minute substances such as hormones, drugs, and vitamins. Or it can use sandwich assay as well. So we can have two platforms just like ELISA. It can be used for competitive assay and sandwich assay. So the difference is that here we are using chemiluminescent dye as label to the antibodies. Okay. So chemiluminescent assay, therefore, is being employed to follow antigen-antibody concentration. So it involves the emission of light caused by a chemical reaction, typically in oxidation reactions. I did mention this a while ago, that decays back to its original state. Okay, so two platforms can be used, either homogeneous or heterogeneous assay. So light emission ranges from a rapid flash of light to a more continuous glow that can last for hours. So different types of instrumentation are necessary for each kind of emission. So there is also a machine that can actually measure the chemiluminescence. Okay, so that ends my lecture for the labeled immunoassay.